So tēnā koutou katoa, no mai ki tēnā koho, ko Neil Gemmell tōku ingoa. So I'm Neil, this is my talk, and I'm very delighted actually for Ange and Manpreet to invite me to give this talk and perhaps give you some insights into some of the things we've been doing over quite a significant period of time now, and also to convey some optimism for the future, because I think we need a lot of that at the moment. So the talk really is about the tools that we have developed and how we've been applying them. Ange has kind of taken a lot of the things I was going to say in the beginning of my talk, and so that means that's 5 minutes I don't have to spend, so it's great. So we live in a country which, you know, at face value looks really quite isolated in compared to the rest of the globe. And of course we have large numbers of unique organisms that occur here but nowhere else. But because of our isolation, we're actually more dependent on trade and commerce than probably many other parts of the world. So many of our goods and services are brought here by boat or plane, and with that comes the risk with those products of biosecurity invaders. And we have had a litany of these over the years. So here's just some of my personal favourites. So we've got various types of mosquitoes, particularly salt marsh mosquitoes, which are coming in predominantly from Australia with things like Ross River virus and various other things. This beastie here, let me just see if this is, which is best. Am I better off doing this, or am I better off doing this? Doing that? You do that? Okay. So just here, this is Styloclava. You'll hear a lot about this little critter. This is the Asian clubbed tunicate, and this occurs really throughout the waters of the world now, but it originated somewhere in the Sea of Japan, Korea, that sort of area. Various types of wasps have been introduced here, various other mammals have been introduced here, and various marine organisms, including this one, Undaria, wakame, is now ubiquitous almost throughout New Zealand. And these species cause damage to our ecosystems, they cause damage to our productive services, and in some instances they also cause damage to our health. And we really don't want them. And the cost of finding these things and then responding to them is reasonably large, even for things that were found to be local and relatively small-scale issues. This is salt marsh mosquito, carries Ross River virus. It turned up around Napier in 1998. Biosecurity New Zealand raced into action and were relatively quick to detect it and find out that it was reasonably localised, but then it took almost a decade to eradicate it. And the cost associated with that eradication, just for this one species in a relatively small locality, $70 million. Now, I don't know about you, but I still think $70 million is quite a large sum of money. And of course, as these problems get larger in scale, the cost of solving them is actually much greater. So it's actually better for us economically if we can detect things and stop them actually getting established, and even better if we can detect them early if they have got established and work out feasible ways to eradicate them. So here's the sort of drum for the entire conference really, is that we need a hero that is going to help us combat these incursive alien species. And that hero needs to be an approach that can decrease the time to detect, monitor and respond to those threats. It needs to be sensitive, it needs to be accurate, low cost, and actually it really needs to be easy, because it needs to be deployable by the people who need to use it at the time that they detect something, or at the time that they are inspecting a shipping container, or at the time that they... I'm not going to give you a talk, Diane. I'd love to give you a talk. Your talk's going to be amazing. It's amazing stuff. So anyway, those early interventions, I'm setting the scene so you can go and, early interventions enhance response, and they reduce those impacts, they reduce costs, they reduce damage. So, of course, I'm going to argue that genomics is indeed that superhero, or at least as applied in this context, it can be a component of the superhero's outfit. And I used in the title this analogy to a Swiss Army knife, and I guess what I'm going to try to do is to illustrate that over the course of a couple of decades, we've gone from the classical Swiss Army knife with, 4 or 5 or maybe 10 features that enables us to tackle particular problems, file your nails, trim your nails, open a bottle of wine, pick out something from your teeth. Never entirely sure about that. Tweezers are actually always very good in these things, I think. Personal experience, to this sort of thing, where, it just got ridiculous, really. Just keep adding blades. How do you hold that thing? And so that's sort of the framework that I want you to take away. And as we navigate through this compendium of approaches that we have used, I'm going to use this framework to describe the biosecurity workflow. So I'm going to talk about discovery. So how do we know what's out there? How do we know that what's out there is something new? Through to surveillance. If we think something is out there and it's starting to spread, how do we document that in a better way? And then ultimately, how do we respond? And I'm going to use, as I triangulate this, a framework of things that we have done in the past, things that we're doing in the present, and also what I hope or aspire for us to do in the future. So let's start with discovery. So this is really basic taxonomy. That's what it really boils down to. Somebody classically would have come with a microorganism or a bug or a piece of plant and asked somebody with expertise, what is this? And of course, somebody with expertise would be able to look at various features, often using microscopy if it was small, and count things or measure things, and then tell you whereabouts in the tree of life this thing probably sits. I mean, some things are fairly obvious, looks like an insect, probably is an insect, although I do appreciate that some people still have trouble with spiders, which are not. But anyway, you'd sit there with your microscope, and I mean, some of us will have done this, counted wing vein patterns on flies, counted numbers of spines on fish, various numbers of gill rakers, and a number of other things that, trust me, you really don't want to do this, okay? But you could, and there are people who have expertise in that space, but actually that expertise is dwindling. And it requires large amounts of materials and exposure to large collections to get good at this. And sadly, we just don't have that capacity anymore. And then I guess about 25, 30 years ago, we started to transition to a space where we really started to describe things more and more based on the DNA sequences that we could find. And many of you will be well aware of this. And there have been massive projects that have tried to utilise these tools with varying degrees of success to barcode life, to describe things using relatively short, simple sequences, and to build phylogenetic trees. And it pretty much works. I mean, it's not, without its risks, but in terms of describing life, it seems to have been reasonably successful. And we've got more and more successful at doing this over the years. This is a paper from just last year, and what its basic shuck and jive is it's looking at beetle diversity for a particular group across relatively large parts of the world. They've collected something in the vicinity of, 30,000 specimens, of which they have then Sanger sequenced 6,500 individuals. So that's a lot of work, right? Sitting there grinding those up and sequencing those. So I pity the poor graduate student that had to do that. They're probably, I hope they're this person. If they're not, they're getting ripped off. But in addition to that, and then they've built a phylogenetic tree with all those, and they've rooted, they've grounded these phylogenetic points with particular species that they've been able to identify. They've done males and females, and then they've done exactly the same thing, really, but with a much reduced data set where they've used predominantly transcriptomics. And I guess the key takeaway from here is that the basic scaffold of the phylogenetic tree that they've produced is basically the same. And so you could argue that you could describe in large scale this diversity using transcriptomic data from, a fraction of the number that you've actually Sanger sequenced. And we still have projects like this that are articulating or trying to find, how good are these new methods versus the things that we are reasonably comfortable with. And I think that's great, but predominantly, and I'm just going to go out on a limb here, predominantly we have found that our new methods actually support the old methods. Now, there'll be exceptions, but I think, we're working in sort of the, 99 studies out of 100 that I see in the journal say that this new technology basically is better than the old technology. So maybe we need to move on a little. And I think we do really need to move on because the pace of change is just staggering. The pace of data acquisition that we are now capable of getting in environmental contexts it just blows my mind. So I'm going to call it the eDNA revolution, and it really starts 20 years ago, with people working on sediments and caves. So this is Esquivelisov's work. I mean, there's probably work earlier than that, particularly for those of you who are interested in below-ground interactions, work on microbes. You'd be going, oh, sort of eDNA. I was doing that in 1996. Or earlier. But, I think starting with soil and then with water in about 2008 and now air last year, we now have the capacity to filter out and gather sequence data on vast quantities of biodiversity relatively simply and easily. And the number of projects that are going on now is really large. So that changes the equation around discovery, I think, because many things from a sequence level will be discovered. They may not be fully described. And we will, rather than doing this side-by-side, taxonomy and sequencing, we will have the sequence and then we'll maybe have to factor back into the taxonomy. And here's some of the things that are going on, right? So these are projects that are happening right now. So we've got the eBioAtlas project, which is trying to look at some of the rarest environments in the world using environmental DNA technologies. We've got the USDA is working both in the West Coast and in the East Coast. You may be forgiven to think that they're not working in the East Coast based on this map, but they actually split them out. And they're doing large numbers of eDNA projects, and they are being stored in a central repository, and they're sharing that information. And our Australian colleagues have the Atlas of Living Australia, where they are plotting the distributions of large numbers of different species, some of which have been described from traditional taxonomic collections and others that are now being described from environmental DNA. This one is actually something that might be of interest to us here in New Zealand, because this is an unwanted visitor to Aotearoa. This is the rainbow skink or the plague skink. This is its distribution in Australia, and of course it's also found here. I did have platypuses, but they don't occur here in New Zealand yet. The invasive platypus, just watch out. But there are many other things. So there is a desire, I think, at national levels and some international levels to work at scale and perhaps to work in a coordinated approach, but it will require significant investment. And I'll get to where we're at shortly. And now, of course, much of that effort is underpinned by work that is ongoing around the world that is gathering data from voucher specimens and developing sequences for specific species. And of course, possibly the first of these was the Barcode of Life Consortium, which I believe is still going. The power of cytochrome oxidase 1 it's pretty impressive, to be fair, but it's not the be-all and end-all. There are other projects like Genome 10K, which is now, I think, 12 years old, so it's now called the Vertebrates Genome Project. But, its point was to try to sequence all living vertebrates. Some of the most conspicuous of our fauna, perhaps not some of the most important of our fauna, but you've just got to start somewhere. And then there have been various other spin-offs from initiatives like that have gone, well, hang on, plants are important too, so we'll do 10,000 of those. Insects are important, and actually they're even more important perhaps, so we'll just do 5,000 of those because, they're only the most specious animals. So why would you aim for I don't know, 10,000? And then you get into projects which are even more ambitious, like the Earth Biogenome Project, the notion that we will sequence all living things on the planet, and related activities like the Darwin Tree of Life Project. I mean, all these things are really trying to do much the same thing. And then some level they coordinate and they work together, but also they compete because there's finite resources. But those comprehensive databases, as they emerge, they're going to empower our genomic discovery even more. So as we go back to our environmental DNA sequences and we go, oh hell, what the hell is this sequence? Over time, we'll gain greater understanding of those data. And actually one of the themes I just want to pick up here, because I'm worried I might forget it, is the power of those data to be reused and repurposed over and over and over again. Unlike, sort of like museum collections. Museum collections are fabulous. You've got to get access to them. These databases are much more portable, much more accessible, and able to be utilised in a variety of different ways, some of which we haven't even remotely thought of. And indeed, the DNAs that we've collected from all these different places also would have value there, but storing and maintaining those becomes a bit of a challenge. So there is a global view that we now have a new lens on the world, and I think we're starting to head that way. In Aotearoa, New Zealand, we also have a number of projects of scale that have started to do these sorts of things. So we have the Open Waters Project, sponsored by EPA, Lake 380 Project, Thousand Springs. Groups like the Marine Biosecurity Toolbox that I'm involved with spend quite a bit of time sampling water and looking for invasive species or better ways to actually detect eDNA from water. We've got wastewater projects, thanks to COVID, that now provide us with a vast quantity of information, not only about what's going on in terms of COVID in our wastewater, but potentially about other disease threats. And increasingly, we also have citizen science projects, high schools that are going out with their classrooms to sample water bodies like this one outside here, do a bit of nanopore sequencing, figure out what's in there and, describe that. Where are those data? Where are they going? Each one of those projects actually, is a couple of $100, $1,000. That information could be useful, even if it hasn't been collected in the most robust way. There might be something that's useful there. So I guess I'm making a plea that we need to try to figure out a way to use all this activity, all this energy in a more positive way. We know this works, okay? We know this works here in New Zealand. It doesn't work as well as if you're in the Northern Hemisphere. Northern Hemisphere reviewers, we don't know the sequences for everything. You guys do. Oh yeah, well you should know what all the fish species are in that lake. And I'm like, yeah, I should, but we don't. You should know all the midge larvae that are in that lake. No, we don't. But unlike you, we haven't been studying it non-stop for 200 years every summer. But sorry, maybe a bit too pointed. If anyone's reviewer 3, I'm gonna have we need to catch up. But we're trying to sequence these things. This is a project from Haturu. This is a little barrier island. It's up in the Hauraki Gulf. It's a relatively small, very protected island on which many lovely species live, including various species of kiwi, some very important insects, and a number of other wonderful things. A project that was led out of the now defunct Alan Wilson Centre went out there to sequence what was present and then compare it with what they could identify using classic biomonitoring. And they found that actually the metabarcodes from the predominant candidates that we use in metabarcoding, 16S, 18S, cytochrome oxidase 1, recovered a vast majority of those species, and I think they described more than 650 taxonomic groups from that work across the island. And so it's a pretty landmark study. But still, there are plenty of things that were missing, and they painstakingly tried to, to get sequences from tax that they collected in traps and then relate those back. But we don't have time for that in all the studies that we're doing. Here's one that I'm involved with, which is on Taranaki Monga, and the purpose of this project is really to bring the Monga back to life. He kawa ora. And it's fabulous, isn't it? Mount Taranaki, it's just absolutely spectacular. And obviously, it's surrounded by a ring of forest. There are native species still present there, but there are also a large number of introduced species. We'd like to remove the introduced species so that native species can flourish. Local iwi are trying to restore species that their mataranga, their local knowledge tells us were there previously, like Theo, the blue duck, and a number of others, robins, kiwi and so forth. And so as we've been empowering some of that work with partnerships with WilderLab, where we go and we help them collect environmental DNA, and then we look at what species are present in areas that are being controlled and predators removed, and also where they are not. And it would be fair to say that the list of taxa and the reliability of those taxa labels for these guys, where they've got a backbone, is pretty good. And all these worm X, something that looks a bit like a stonefly, and things like that. So we could be doing a little bit better there. So here's my sort of take-home message one, which is, why don't we have a bioinventory for New Zealand? I mean, we should have a bioinventory for New Zealand. We currently have piecemeal efforts that have been probably driven mostly by local context or by taxonomic context. I'm involved with a piece of work that Libby Liggins has been leading, an environment link grant, which is going to look for to develop a bioatlas, a bioinventory, I guess, for insects and for some fishes, but there are many, many other gaps. And so if there's one simple thing that we could do, it would be to go out there and try to sequence all the species in Aotearoa, New Zealand, and apparently there are 80,000 endemic ones. Now, it sounds like quite a big number, but, you know, some poor graduate student just sequenced 6,500 beetles. So we could do this. It might take more than one graduate student, but we could do this. And the other thing that I'm really passionate about is if we can get that sort of inventory together, then the future prospects for this technology are vast. Now, how many of you have used iNaturalist? About half of you? Yeah, so it's a pretty fun tool, isn't it? You know, this is a bug I don't know, or this is a plant I don't know. I'm really, really bad on plants. So apologies for those of you who are botanically inclined. I am going to ignore that. Predominantly. Although iNaturalist is great, I can go, what's this leaf? What's this plant? So, you know, now you can take that photo, a group of experts will look at it, give you a consensus view on what that should be, and even iNaturalist will try to do some sort of pattern matching for you. Well, I'm proposing we have DNA Naturalist. I've even got the logo and the name. This is me, this is mine, I invented that. And, you know, at the moment, a flongle or something like that from Nanopore, and I don't have any shares in the company, costs you whatever it is, less than $500, isn't it? 300? 300 or 400? 300 or 400, which, you know, is not an insignificant sum of money. But I'm also mindful that USB sticks used to cost real money as opposed to being given out as part of a conference promotion. You know, you go to the store, you actually buy these things, you pay your hard-earned cash. Pocket calculators used to be expensive. I'm really that old. You know, and various other things. I think this is going to commodify. I think it's going to get really cheap. I hope. I really hope it does. And then you can put it onto your favourite device, and just for giggles, we could go out at lunchtime and we could sequence some stuff because that's part of our daily routine. It's what we do. We've decided that we've taken an interest in this particular piece of water and that we actually wanted to understand it over time. Just as the same way some of us have weather gauges and various other things at home. And then you integrate it with those databases and bang, 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 you know, here is the information. And then just to make it useful, it goes up into the cloud so other people can use it. That's the discovery framework that I'm thinking of. Well, that's great. I've taken more than half my time to do my first point. So, just right on time. That's okay, I'll just cut some stuff out. But I think the pace is good, I'm enjoying this. So let's move on to surveillance. So obviously if we've discovered something, I mean, and actually these two things overlap. because surveillance could be ongoing surveillance to stop incursions at borders. So we want to identify those incursive species. We want to identify, once we've found them, where the hell did they come from so we can stop more coming in. And if they're here, we want to know how they move about so we can stop them spreading like cane toads in Australia. I'm not going to give up your talk either, Lee, okay? Not going to do that. Just admiring the mahi. All right, so point of entry focused is what surveillance needs to be. You know, but it's predominantly still identification in a laboratory, hours or days after interception. You know, particularly if we're going to use taxonomic focus, that's going to require going to somebody who has the expertise and then being trialled. Now, you could probably take a photo with your mobile phone and then send it to iNaturalist or iExpert or iTaxonomist or whatever you want to call it, and maybe that'll be a bit faster. But they're still probably going to want to see the physical specimen because #reasons. So what we really want is a desire for a real-time on-site identification that needs to be fast, it needs to be accurate, it needs to be reliable, it needs to be portable, it needs to be easy to use, it needs to be low cost, it needs to be high throughput, it needs to wear its undies on the outside. It needs to be good. So I've been working on this problem with the Marine Biosecurity Toolbox now for quite a few, well, 3 or 4 years now. And actually two of the things that we've been trying to tackle are this, simplifying sampling, particularly of water samples, and developing point-of-need capabilities. This is a little bit awkward, this sort of over-the-shoulder thing. So classically, those of you who have sampled eDNA from water will know that requires good calluses on the hands or various other devices to push large quantities of water through relatively small filters using syringes, or if you're really lucky, you can go out and buy a $15,000 piece of kit that'll do it for you. Most of us don't. And that has some limitations. The amount of water that you sample and how turbid it is will very much determine the sorts of samples that you actually collect. So an alternative is to use what we call passive samplers. So you're basically just putting out some sort of positively charged membrane into the environment and hoping that the eDNA will interact with it over time. And the good news is it does. And it does pretty well. And there are groups like ours that have shown this reasonably well. We could refine this further by developing surfaces that are more amiable for DNA to hit and bind to. And that's a piece of work that we're currently engaged in. The other thing that we've got into was this idea that there are organisms out there that are constantly sampling their environment because that's how they live. They're filtering things from the environment to eat, and you could then check their gills or their stomach contents as a way to look at what species are present. And that's been reasonably successful too. And I'll show you some data for that shortly. And then we've been developing point of need using nanopore technologies and extractions in fields to look for various things. And lots of that work's been led by Joanne Stanton at Otago, and she's had quite a degree of success working on detections of cassava viruses and various other things in Africa using these technologies. This is where I really want us to be. If it's a single species that we're interested in, some form of lateral flow device, similar to what we're using for our tests for COVID and so forth, but this is linked to sort of CRISPR-based detection technologies. We haven't quite got there yet. So let me just tell you a little bit about our sponge work. So we've tried a variety of different marine organisms. We've tried mussels, we've tried barnacles, we've tried some other things. They kind of work. Sponges work really, really, really well. Don't ask me what kind of marine sponge, because we'll need a taxonomist to figure that out. And they do most of their analyses using spicules, but there's about 3 different species that we've collected over time. And this is just some data, and it's not optimised yet, but this is using our extractive eDNA procedures, filtering about 2 litres of water. This is a diver survey, and this is with sponges. And sponges we've actually got better at now. But the basic take-home message here is that just taking a little piece of sponge and extracting it does tell us most of the fish species, does detect most of the fish species that we find in a particular locality. And the eDNA yield is very similar to the commercial filters. So we've just got a piece of work that hopefully is about to be accepted. that describes that. That paper is on bioarchive for anyone who's interested, and the lead author is Jan Yunin, who's done most of the work in that area. If you want to know more about it, just ask me. I'm going to segue now to some old work, which is on this Asian club tunicate, the Asian Ascidian. I'm going to use this as an example of a few different things around surveillance. So this is a global invader. It's found almost all across the globe now. It started out in Asia, hit North America about the 1920s, moved through the Panama Canal, through into Europe in the 1950s, 70s, then into Australia in 1978, and apparently New Zealand in 2005, but I think actually a lot earlier than we ever detected it. Here's some work from Sharon Goldstein and colleagues, and here's New Zealand. This is circa about 2006 we're sampling, and the mitochondrial diversity in New Zealand is astronomically high. In fact, actually higher than in the native range, which is here. And this is Auckland, and this is Lyttelton, and you can see that the mitochondrial haplotypes that are shared between those sites are actually very, very low. And so this actually tells us a number of different things. So first, it is obvious that we have incursions from multiple sources, and I'm going to go out on a limb and say they didn't all just arrive in 2005. And the other thing is that the shipping that has brought these in is independent of these two locations. So there's clearly migrations, connections with Auckland shipping, which are different from those in Lyttelton, and there seems to be very little in the way to suggest that there is a lot of migration between these two sites, although there are a few shared haplotypes, obviously. And actually, serendipitously, we got a detection in 2006 in Opua on this vessel here, fabulous-looking schooner called the Artaka Thompson, and it had Styloclava on the outside of the hull, and that single boat had 3 different haplotypes, 3 different mitochondrial variants. So already we can see from the genetics that we're getting a sense that these incursions are happening frequently, that when they happen, they are bringing in multiple haplotypes, and that the actual pressure in terms of new variants into the environment is actually quite high. But that was a lot of work to do that. I mean, a phenomenal amount of work. Sharon had to go up and sample Styloclava in Auckland, and hours in the water collecting these things, and then back to the lab and extracting and various other things. So that's a full postdoctoral piece of postdoctoral research, 2-3 years work. Maybe we could have done it faster. Maybe we could have done it a lot faster. And this is where I think surveillance is heading. And yet, and this annoys me, actually. This annoys me. 2007, global ocean sampling expedition. So this is Craig Venter sailing his yacht around the world, dropping buckets over the side, collecting some water, taking them back, and then metagenomically sequencing them at a time when that was not cheap. Still isn't. Okay? Collecting phenomenal amounts of data. Actually, not so phenomenal anymore. So I first learnt about this through a project called, where I started to get into sort of advanced computing and sharing data and those sorts of things. A project called BestGrid, which ultimately became Nessie, our science research infrastructure. But this project called CAMERA, it's got a very long name, circa 2007, was collecting all those data. What were those data? Well, they were 71 metagenomic studies, about 800 data sets, and 48 billion base pairs of data, or 120 million reads at the time. Now, I'm not going to talk much about Loch Ness, but our simple Loch Ness project is 6 times bigger than that. And that was a big project 4 years ago, but it's not a big project now. So the sorts of data that we're talking about interrogating, you probably have in your own labs, for those of you who are doing eDNA. But if you search those data, we found Stiella in those samples taken from California, Europe, and a number of other locations. This is going back, 2009, 2010. So there was an opportunity already from those surveillance data to make detections in some contexts. And imagine if we could scale that up. Imagine if we could scale that up, then we can detect things before they become problems, or we can detect them as we respond to them and see whether or not we are making them less of a problem. This is a project that we're actively engaged in at the moment. This is data from Kristen Fernandez, who's just started as a postdoc with me in Otago, working with Sean Wilkinson and Mike Bunce. The working title for this is Wildermap, but I think we're going to have to change that. We can't, yeah. But this is the eDNA data we currently have for Aotearoa in New Zealand that Wilder Lab are sitting on. There are over 3,000 sites, each of which has generally been sequenced with at least 4, sometimes 8 metabarcodes that span insects, fish, mammals, a bunch of other things, plants, don't forget plants. And so we are interrogating these data to see what the current distributions of things that we might care about are. So this is possums mapped across those data, and this is rats. And hypothetically, insert your favourite species here: stoats, weasels, ferrets. Good to see you, Andrew. You know, and then we've got a really quite powerful resource for looking at where things currently are and asking the question, are we targeting our efforts appropriately in terms of the control strategies that we're using? So this could be used to inform strategies like Predator-Free 2050, which is the desire to remove New Zealand of some invasive mammals, but not necessarily all, by 2050. And of course, the Department of Conservation is actively engaged in various control measures to try to reduce the damage of many of those species. We keep doing this, we can see when we're making a difference. We're not necessarily going to get to an absolute quantification, but we should be able to see when we've eradicated things from particular regions. So here's my take-home message too. We could do this. Okay, so that Wilder Lab project is driven predominantly by citizen scientists or regional scientists who are going out sequencing things. But I think we could be sequencing everything, everywhere, all of the time. It's a bit of a mantra with me now, those of you who have heard me talk about this before. And this is how it works. We are finicky about our water. We are finicky about our air. We are filtering all that stuff all the time. In this building, I think, we are probably filtering the air that I am exhaling and you're exhaling. That's collecting stuff. There are filters. People get paid to change these things. We could be collecting those, we could be sequencing those, and we could be analysing what is present in a particular space at a given time. This could be phenomenally important for public health, sitting on that plane, sitting in this room, sitting in a restaurant, sitting in a bus, drinking water from the tap, flushing the contents of that water down the drain later through other measures. All these things are biological materials that we could be investigating to look at diseases, to look at invasive species, to look at biodiversity. And the rationale for this would be, well, you know, that's what I'm told we're spending each year on or bioinvasives, or what they're costing us. So you could argue that if we used a fraction of that money, that would be money well spent. Here's a case study for New Zealand. And Ange and I will need to sit there and figure out what our data sources are in case we're talking at cross purposes. But it really doesn't matter. These are big numbers, right? They're just big numbers. So this is biosecurity in New Zealand. I'm told the economic cost of pests in New Zealand are about 9 billion per annum. We spend about 1.5 billion per annum to protect ourselves against those. In health, a level 4 lockdown cost us $290 million a day. And seasonal illnesses, and gee, haven't we had a lot of those recently? COVID notwithstanding. I don't know about you, but my workplace has just been, you know, oh, and I'm off with the flu now because I haven't had it for 3 years and it's a novel thing. 1.26 billion per annum. Sorry, there should be a billion And then in biosecurity, the things that really drive our biosecurity are economic impacts. Mycoplasma bovis, which was detected, 5 years or 6 years ago now, is going to cost us almost $1 billion to eradicate. And yet, if we'd had on-farm surveillance across the board, we'd have picked up the herds that had that pretty quickly. Foot and mouth, which I know Australia's worried about, and we should be too, in fact I know we are, if we got that, and it was an outbreak that was restricted only to the North Island of New Zealand, MPI says it's going to cost us $15 billion in lost earnings, and here's the kicker, our GDP as a nation would be $24 billion lower every year for the next 5 years. That's massive. So we might want to spend, I don't know, 100 million, 200, 800 million dollars doing this, just to safeguard ourselves, I think. And we know we can do it. We can do this. The pandemic has delivered many things. It's delivered some pretty spectacular results in terms of the way we monitor things. This is COVID-19 and wastewater here in Waikato. So, up to last night. And, you know, that's real-time information or near-time information that is useful. Globally, there are 152 dashboards like that, collecting information as people that you can use to make decisions. Wouldn't it be nice to be able to make those sorts of decisions about other things? We could, for example, look at antimicrobial resistance in sewage. And I was involved with this study, and that's actually how I first got involved in sewage. We went and we collected some samples here at Tahuna and Dunedin, which turned out to be, along with Australia, amongst the lowest levels of antimicrobial resistance that were detected across the planet. But wouldn't it be nice to look at that over time? Wouldn't it be nice to be able to go back and say, well, actually, we stopped using antibiotics in farm animals in these places, and this is the response that we saw in terms of total antimicrobial resistance in the in the environment. And just, you know, spoiler alert, there's always going to be some level of antimicrobial resistance, right? But it's just a little bit rampant. How am I doing for time? Oh, I've got about 10 minutes. Okay. So, I think New Zealand should be bold and brave, and we should take projects like the ones that we already have through WorldLab and expand on those and augment that work. Add the data sets that we're collecting to a national collective so that we can better understand what's going on in terms of diseases and infectious disease vectors, species that do economic and ecological damage. And dare I say it, as we are monitoring these things, we can also be monitoring these. And I find it frustrating, having worked in conservation now for 30-odd years, it feels like. Yes, I am that old. And every day I feel it. That was a bit of a joke. You want me to go, oh. Surely not. But, you know, we still have these arguments in this country of, oh, well, if you control pests, what's happening? You know, what's the value of controlling pests? Deer are not a problem. Possums are not a problem. Well, yes, no, they are. I can show you the data that say that when they are present, you do not have these things that I value, like, I don't know, these guys. So I think one of the things we can do is we can help to build that knowledge base and show in near real time, as we control these pest species, that we get biodiversity gains. And it's not always in the things that you would think are conspicuous. You know, we're not going to get a boom of takahe overnight, but we will see, hopefully, improvements in tussock grass regeneration and various other things that they may be dependent on, or various other insects which will respond much more rapidly. Wouldn't that be neat to be able to show that in real time, just like we show COVID data in real time, near real time? But there are some real challenges associated with this. So obviously our analyses are only as good as the databases we have, and frankly our databases aren't that good at the moment. Diane will be able to tell us about all this. You need to have standardisation of approach, because if we're all collecting stuff in different ways, that can be a bit of a problem. I mean, it's probably better to have some data collected in a variety of weird ways than no data, but if we really want to analyse it in an integrated way, we should standardise it. In this country, and indeed I think globally, data sovereignty and the rights of Indigenous communities need to be considered, and how we use those data, how those data are stored and how they are accessed are really important issues that we need to tackle. And there's been a lot of work here at the University of Waikato from people like Maui Hudson on cultural biolabels and a number of other things, which I think are important tools that may enable us to move forward with some level of confidence. But we really need to nationally engage on these issues. And again, because I'm old, I've been saying this feels like for quite a while now. I'm going to take just a few minutes to talk about response. So clearly, when we have information, that information can inform our response. How we responded to COVID in Aotearoa, New Zealand, was informed by information. We could see, those of us that were paying far too much attention to Twitter, before it was owned by Elon Musk, that we were about 3 weeks behind the rest of the world in terms of an eruption of COVID in this country. We could see what was happening in the UK and Ireland, what had happened in Italy, and we closed our borders, maybe 3 days a week after I wanted to, but I wasn't in charge yet. No, I really don't want to be. Really don't want to be in charge. But I was pleased that that was a science-informed response, and I know it was distressing for many, but I think we did the right thing at the right time. And that was informed by data. And all these things could have been informed by data. Mycobacteria, the other one. Mycoplasma? Yes. Those sorts of diseases, Mycobacterium tuberculosis is also a problem, Johne's disease. But, diseases of agriculture, if we could detect them early, that would be great. That would stop things from dying or us having to kill many things. And kakapo, they've had diseases, aspergillosis. We're currently monitoring for that in near real time to see if this is going to be a problem in current breeding seasons. Obviously, SARS and then SARS-CoV-2, avian flu, all these things we can monitor. And what we really want to be able to do is actually get a bit more predictive. And to be a bit more predictive, we need to know not only when they're present, but what the source and patterns of spread are. If we want to build good tools to control them, we need to know weaknesses, like, for example, the spike protein to enter a cell, so we can build vaccines around that. That's a pretty good weakness to know about. And we need to know how they evolve, so it's frustrating to me as an evolutionary biologist that for the last year we've been calling a virus, which is now very different from what it used to be, the same thing. Okay. Quickly cover a couple of these points, and apologies for starting to get very close to going over time. This South Georgia Island, this is actually one of the first projects I was involved in where we actually used DNA, genetic information, to guide a eradication program. So the question was, could we find any evidence that populations of rats around South Georgia were moving, whether there was gene flow between those and therefore they were not isolated populations, or were they indeed isolated populations, each of which could be amenable to eradication? In other words, if they dropped poison there, they got rid of the rats, they weren't going to be recolonised by rats from another location. And I apologise for this figure, it's not very clear, but there is a fjord here and there are glaciers here and there are populations of rats here and populations of rats here that we explored, and we showed that there was indeed absolutely no gene flow between those two populations. They had a large number of fixed loci across, I think at the time, 24 microsatellites, which I thought sounded like a lot. It was the advantages of working on something that had a genome. So then they started at that particular point in about 2011. They dropped poison, they eradicated rats from there, and then they subsequently extended that poison eradication through the southeastern part of South Georgia and then up through this northern part. They dropped 300 metric tonnes of 1080 on the island, cost them $13 million, it took them 5 years, but rats are gone from South Georgia Island. It's probably one of the biggest eradication programs that have been undertaken. And the consequences of the eradication are positive, you know, for seabirds, for insects, for vegetation. I think I might start to sum up here. So I won't go into too much of the weeds on this. I know many of you will talk about this as we go forward. But clearly, these genomic sequences are really powerful for providing a framework in which we can undertake population genomic analyses, but they're also great for looking at what are the weaknesses of various species. So we've been involved in sequencing the shiprat genome, which surprisingly hadn't been done at the time we'd done it. We've worked on a variety of weevil species, and in those instances we've not just done the genome, but also the microbiome to see if we can find inherent weaknesses. We've just finished the possum genome, and that paper's coming together now. Well, actually, we finished a few years ago. We've finally analysed it. But, I mean, there's already susceptibilities that we're aware of. The way they use their water balance mechanisms are different from other mammals. Obviously, they have marsupials, have different types of milk. You could target that, but, you know, nobody really wants to have starving, dying joeys all around. They're very susceptible to vitamin D toxicity and a number of other things that you could use. This is not my work, but it is about Styloclava, which I've spoken about before. This is the genome sequence from Styloclava, published 2 years ago. They have about 547 unique genes that they've identified, and in that gene complex, they find a large number of heat shock proteins and also cold shock proteins, and they claim they're horizontally transferred, but I'm going to have to look at that in a little bit more detail because that's always a very... I'm always suspicious about those sorts of claims. But the point is they have a large number of these genes which enable them to deal with both heat and cold, and that may explain why they literally are almost everywhere. If we could make them less adaptable, that would be good. I could spend more time talking about different ways to control things using GMOs and gene drives, engineered genetic incompatibilities and other things, but I think I shan't. I'm now going to show how bad my time management is by skipping through large numbers of slides to finish with genomics is a fabulous tool. It really is. Or at least I think it is. I mean, I was debating whether I wanted to show you another Swiss Army knife with many, many facets or just a very adaptable hammer. Because maybe it's that. Maybe it is just the one tool, but the way we wield it with various levels of subtlety is what makes it different. Or a Dremel. Anyone know what a Dremel is? Which cuts and drills, and that's about it. But it sounds really good. I'm going to finish with this, because I think this is a discussion point. I don't know what it is about biologists, but we're done with money. Physicists, astronomers, they're really good with money. You know, hey, I want to study the origins of matter. Sure, have $10 billion. I want to understand the origins of the universe. How much is that going to cost? $10 billion? Done. I want to understand the genetic fabric of being human. How much do you need? $2.2 billion? 2.7? And that's just, you know, we need to be more ambitious. And there are, I mean, you know, let's not even talk about human health. And that was on the back of, you know, human health, which is what? Probably apart from military defence, probably the most expensive thing in the world. That's phenomenal. This is what it costs us in terms of bioinvasives. Could we not make an argument that instead of looking out to space or looking into various types of colliding matter, that to understand all of life, an investment of, let's say, 10, 20 billion dollars globally might actually be a very, very wise investment for the future. I'm going to finish just to acknowledge that I almost do none of this work myself these days, and that very large numbers of people have been involved, and I'm just sort of dispersing them through the frames of marine biosecurity, terrestrial biosecurity and biocontrol, which I actually skipped over. But I'm just very grateful to these people for working with me over many years. So thank you. Thank you for your attention. Do we have time for questions, Manpreet?
We have 5 minutes. We have about 5 minutes for questions. And first of all, thanks, Neil, for this fantastic talk with a broad range of topics. And hopefully there's lots of questions from the audience. And also, we would like to welcome our Zoom participants because this event is being live streamed and recorded. And so if there's any questions from the Zoom participants, we'll try and take those as well.
Where are they coming?
Right behind you.
Oh. That's what the back of my head looks like.
Question.
You said at the start that the superhero thing needs to be easy. And, or like the, yeah. And I would say that maybe it's still at the moment, without training, with training genomics is quite hard.
Yeah. Yeah, I think pipelines are really key to this. So, you know, I mean, let's take iNaturalist. There's a lot of very sophisticated software that's cracking on behind there to take your image and then figure out what it probably is. We don't need to worry about that, right? I don't have to be an image processing expert to figure that out. We need DNA to be to work in a similar way. I mean, you do a Google search. There's a shit ton of stuff going on behind the scenes. You know, 0.1 of a second later, there's your list of things. That's what we need, I think. Obviously, got a ways to go. But, you know, there are databases, you know, and instances that you can use. I mean, so Kraken 2 and Kma-based analyses, they can be reasonably fast. You can break that stuff down reasonably quickly. So I'm optimistic that we will find solutions that will enable people to take what is alien to them. You know, I've collected a sequence. They don't even know what I need to know what the sequence is. They don't need to see, you know, the sorts of things that old-fashioned people like us might want to see, like chromatograms and strings of DNA sequences. They don't need that. They can all be happening in the background. You need to be able to punch into that to know that it's doing the right thing, but from a user base, it doesn't need to look like that. So yeah, I think that could be where we get a transformation. But first, there needs to be a demand, and the demand needs to be driven by the price point of instrumentation. And also, just to point out, sorry, if we were to move down this process of us all sequencing everything everywhere, the game would change profoundly for the sequencing providers, because then, you know, at the moment they control the price point, and it's driven. That price point is driven predominantly by what the healthcare sector is willing to spend. If we are talking about sequencing which is at 100, 1,000, 10,000 times what we're currently doing, then environmental monitoring becomes the main driver of the need for sequencing, so we should be able to set the price point. If governments are investing in this, governments will arrange pricing that suits them. So the entire thing could just flip from being relatively rare, high value stuff to lower value commodities. I mean, let's be honest, you know, what's in those tubes is just chemicals. And what's in my can of Coke is just chemicals. And they probably don't differ that much, except that one is very rare and the other is very common. And a can of Coke is now cheaper than milk, so go figure. All right, that was probably far too long. Lee? Billion. Did you? Oh, I thought I heard million. I felt like I was in one of those sort of Bond movies.
Yeah, I'm just wondering if you have any thoughts about ways to increase inclusivity so that some of these initiatives within product closed. How do we do this on that scale so that we all work together?
Yeah, I think that's possibly the biggest challenge. I think it needs to be led government to government. I mean, it could start scientist to scientist, but the problem is that there's always going to be somebody who feels that they're missing out. I mean, I guess two different models: large-scale government investment, or a coalition of the willing, with people paying what they can to be part of something big. I mean, the genome projects that I've been involved with have often worked that way. Somebody may have the resources to pony up the money to get the genome sequence, but they don't have the resources to pay everybody to do all the analysis. Everybody brings something to the table, whether it's in kind or direct. Maybe that's the way to do it. Maybe you've got networks of people who are willing to go out and take the devices into the community and do the sequencing. That's a time and money equation, as much as the processing in a lab. So, I mean, maybe it starts organically from that level, Lee, with a really kind of like a crowd-funded sort of or science lab team lab or something. I don't know. Collaboration. It's an online collaboration of people willing to be part of something, and then ask them to make a modest investment if they can, and if they can't, well, how else are they going to contribute? Maybe that's the way to start. Have I convinced you? Yeah, I would too. I mean, this is, I think, these sorts of conferences, these sorts of conversations are opportunities to first ask the question and then see what the response is. And the response I'm getting is maybe positive, but I mean, you can be sceptical for sure. If it's a pipe dream, maybe it is, maybe it's not. Got to start somewhere. And actually, we've started. We've got a lot of data. Here, Di.
So, are you aware of the initiative coming out of the Simplex lab in Switzerland, which is they're proposing
So this is Christie.
Yes, and those Franklin tokens. So basically, those fungible tokens that then what it will entail is the minting of these tokens that then have a monetary value associated with them every time you sort of submit a sample. It's quite a complex. There's a white paper on it.
I'm going to have a look at that. No,
Yeah.
I mean, I know Christie and some of her work and some of her thinking in this space, but that's actually new territory for me.
It's very new. It's only just come out in the end of last month.
Okay.
And it is something that is sparking a lot of interest in the eDNA community, and particularly all these eDNA consortia around the world are now looking at this as a way of then bringing them together. But the thing is about that accessibility, and also for countries where the resources are super limited, and often those are the countries that we really need to be investing much more in terms of their biodiversity monitoring, and it's in a way of enabling that to happen. So I think that's an interesting thing, and possibly something that we can discuss in a conversation when we get to that on Wednesday.
Yeah, that sounds really good to me. I think we need to find ways to do this. And we need to find ways to share our data, and we need to find ways to share those data securely. I think in New Zealand, we need to be mindful that there are concerns about how those data could be used for bioprospecting, or how they might actually result in some level of additional exploitation, for example. And this will be true in many other parts of the world where you've got endangered species. And actually the last thing we want to do is be able to tell animal traffickers exactly where to go to pick up a species which is critically endangered. So there are, yes, so in some instances we want to be open, but at the same time we need to find ways to also be aware of risks. So I think it's going to be a tricky one to navigate, but I am optimistic that we could do this, and actually that we should.
I mean, we do this in human health, right, where the data is very secure, and yet they're able to share.
Yeah, I mean, some are more, some share more than others. So obviously some of the larger scale studies like the UK Biobank and those sorts of things, those data are available. You sign a declaration saying I'd like to use these as the purpose, you get approval, and there you go. And there's others that you can just access already. And then there's others that even when it's published you can't get hold of. So it's kind of variable, isn't it?